Hello everyone, myself Rajveer Singh Shekhawat, and the title of this presentation is Identification of Common Prognostic of Biomarkers for Type 2 Diabetes and Cancer using Expression and Network Analysis. Coming to the introduction part, the diabetes and cancer are chronic diseases that have imposed a global health threat. These two diseases have common risk factors. The increase in incidence and mortality associated to these two diseases is also quite high and a point to discuss. In the past, several epidemiological studies have already suggested that type 2 diabetes as a condition increases the risk of several cancer types, that is liver, pancreatic, breast, and endometrial. But despite these studies and meta-analysis studies, the link is not that quite clear. And also, the diagnosis of symptoms at an initial stage is quite difficult. So there needs to be a systematic genetic linkage study that needs to be performed to identify this linkage. So coming to the methodology part, we first collected the microarray data from the PBMC sample of patient as well as healthy individuals. Then we processed and filtered that data in our software. Then we performed the differential expression analysis, and then we found out the common differentially expressed genes. The FDR value threshold was 0.05, and for the log FC value it was 0.5. And then from these common tags, we performed the enrichment analysis using the gene coders software, and we found out the significant CAG pathways and gene ontology biological process. The FDR value was 0.05. Then from these common tags, we also performed the protein-protein interaction network analysis in the cytoscape, and we integrated that enrichment analysis results in this network. And then we found out the hub genes using the cytohub plugin. Then we perform the survival analysis and to look whether these subgenes are associated with poor survival in cancer patients. And then to validate these subgenes, we took a validation dataset and performed the validation step that we will discuss further. So we collected these data from the Gene Express Omnibus. These all are publicly available datasets, and the accession ID is provided here. So we collected data for type 2 diabetes, pancreatic cancer, liver cancer, breast cancer, and their respective healthy, and also validation dataset for validating our hub genes that was for type 2 diabetes patient and the corresponding healthy individuals. So coming to the results part, the shared genetic association which was investigated and identified between type 2 diabetes and the three cancer type, pancreatic, liver, and breast cancer. So the first column common is showing you the common number of genes between the type 2 diabetes and the three cancers. The first filter criteria was statistical significance, that is the FDR, and after that the number of common genes are shown here. The second filter is biological significance, that means the log to FC filter. After that the number of common genes are shown here. The third filter is relative distance, which means that the gene that is upregulated in type 2 diabetes must be upregulated in the cancer patient as well. And if the gene is downregulated in type 2 diabetes, then the gene must be downregulated in the cancer patient as well. The function enrichment analysis identified through the common genes showed enrichment of some important CAG pathways and geobiological process that we will see in the next slide. And also the protein-protein interaction network analysis revealed certain important genes that were critical for the network. So on the left hand side, you can see the protein-protein interaction network for type 2 diabetes and pancreatic cancer. The nodes that are red in color are upregulated. The nodes that are blue in color are downregulated. The color intensity is showing you the log fold change, and the size of the node is representing the degree of that node. That is to how many different nodes it is. The single node is connected. The colored ring around the node is showing you the gene ontology, biological process, and CAG pathway that this gene is involved in on unrestrained. So these are the pathways or processes that are enriched in this network, and these all pathways and processes are important for type 2 diabetes and pancreatic cancer. Some of these pathways are already reported in previous studies that are linked to type 2 diabetes and pancreatic cancer. So after the protein-protein interaction network analysis, the next step was to find out the common hub genes using the cytoba plugin. We found out that 7 hub genes were found in type 2 diabetes and pancreatic cancer network, 6 in type 2 diabetes and liver cancer, and 5 in type 2 diabetes versus breast cancer. And one gene was common in these two conditions, that is TGF beta 1. It was common in PC-T2DM and LC-T2DM network, and the expression of these total 17 hub genes is shown in these bar plots. You can see, and the name of the gene is mentioned in the x-axis. And we also performed survival analysis, and we found out that 4 hub genes out of total 17 were strongly associated with poor survival in patients, and the FDR value is also given. So in conclusion, our analysis provided insight into biological and molecular events that could link the type 2 diabetes and the 3 cancer types. The enrichment analysis highlighted the cell cycle events, immune system process, cell morphogenesis, gene expression, and metabolism are that were highly enriched, and these all are related in somehow to type 2 diabetes and the cancer. The validation of 12 out of 17 hub genes could be performed from that additional dataset that we mentioned in the starting, and these identified genetic markers hold the potential to predict the chances of cancer onset in the type 2 diabetes patients. So I would like to acknowledge my co-authors in this study and also my lab members. This is the lab that I am currently associated with in IIT Jodhpur. These are the references. So this study is already published in scientific reports. You can check this paper. Thank you. Any questions?